Okay, hi there, everyone. We're going to get started shortly with our weekly Q&A, and I already saw a question come through. Yes, participants, we currently have your microphones disabled. If you have questions, go ahead and type them in the chat at any time. We do have a little bit more of a directed content this week, talking about our PCR product technology. So we want to present that first, but please enter questions as you think of them. Additionally, I will have kind of our standard overview deck for questions after at the conclusion of this presentation. So just want to do some introductions. Those of you that haven't been on one of these calls before, I'm Andrew Anfora. I am the FAS manager for Universal Sequencing. Additionally, Tom, if you want to, I guess everyone else on the call from UST, if you want to do intros.
This is Tom Chen, CSO of the company.
This is Ivan Bassett. I'm the R&D director.
And David Nordell, I'm the inside sales manager.
Okay, so I will turn it over to Ivan, and wants to describe a new application that we've released an application note for, and we're really excited. We know we've been getting, I get a lot of questions from new customers about Amplicons, so here's a great little story we wanted to tell you, and then open it up to Q&A.
Okay, thank you, Andrew. So do you mind to keep sharing the slides, Andrew?
You can go ahead and share.
It's you? Okay.
So go ahead and share your slides. Yeah.
It's gonna be a problem. It's maybe if you guys share the slides.
Okay, go ahead then.
Okay, so in the next 6 slides, we will show how the haplotype phasing of PCR products is possible with a few modifications in the protocol of the TELL-Seq library prep kit. So we move to the next slide. So in the current user guide, we provide guidelines to process samples with DNA sizes ranging from the entire large genome to targeted high molecular weight fragments, ranging between 50 and 200 kilobases. So the new user guide that will be available within a few days will include modifications that enable the processing of PCR products, as Andrew said, between 1 and 20 kb. These modifications significantly increase the repertoire of targeted phasing applications. And this talk is going to summarize our results using this modified version of the user guide. So next slide, please. Okay, so in essence, there are only 5 modifications to the standard protocol, and the processing time is the same. So the modifications are shown in this slide are 5. So in one TELL-Seq reaction, the input DNA in this case will be 80 picograms. That can be either a pool of DNA fragments or it could be a single fragment. If it's a single fragment, then we recommend actually adding a competitor DNA. Normally, it could be E. coli genomic DNA or lambda phage genomic DNA. Then the additional modifications are that the resuspension volume after bit washes, it will be 40 microliters. And then from there, we just take 1 microliter. So again, these are slightly small modifications from the standard protocol, but they're important for the processing of PCR. And then other modifications are the number of cycles in the PCR, it's slightly higher. And then the elution is a volume that is smaller than usual. So we can move to the next slide. Okay, so we have validated these modifications on the user guide with 2 examples that I want to show today. The first example is the case of a composite mutation. So as a reminder, composite mutations are those that when you can find more than 1 mutation in the same gene, in the same sample, basically in the same cell. So recently it was reported that actually composite mutations are very common or frequent in cancer. Approximately a quarter of all the tumors have composite mutations. And for our testing purposes, we have focused on one particular example, which is the case of the PIK3CA gene. So in breast cancer in particular, it has been reported more than 10% of composite mutations in tumors. So then we focus on one particular case of a double PIK3CA mutation found in the cell line HCC202. So we use this cell line for our test. So those two mutations are separated almost 1 kb, one from the other, but they are located in different alleles, in different chromosomal copies. So they are in what is referred to as intrans configuration. So this is a perfect example for phasing. It happens that this cell line also has a single nucleotide variant a little bit upstream of one of the mutations, which is in cis with the first mutation and in trans with the second mutation. So again, this is a perfect example to see how our user guide, the new user guide, actually works with phasing of PCR products. So if we move to the next slide, okay, the second example I'm going to show results about is one of a very common approach in research, which is a genome-wide association studies. So as probably everybody knows here, so genome-wide association studies where you have a hit, a positive, a nucleotide, single nucleotide variant that is associated with disease that emerges as a hit. Normally, it doesn't do it as a single hit, but as a cluster of hits. So then this provides an opportunity for phasing, because what we want to know is how those different single nucleotide variants actually phase between them, because sometimes that risk may be associated with one particular haplotype, but not with specifically the presence of the single nucleotide variants. So for this example, we took the case of GWAS that was reported in the study of life-threatening heart arrhythmia. And then they identified a few single nucleotide variants associated with the risk for this condition. And then we took a region of 13 kb that includes these single nucleotide variants, and then also shorter versions or shorter fragments of that region for the purposes of this test. I didn't mention in the previous slide that also the total fragment in that case was 3.4 kilobases, but for the purpose of this test, we also use a shorter fragment of 1.8 kilobases. In the previous example, the input DNA was PCR amplified from cDNA. In this second example, the input DNA is PCR amplified from genomic DNA. So if we move to the next slide, I will show you. This is the summary of all the results. It's not my intention to go over all the specific details, but just what I'm showing here is 58 tests, and they are based on different conditions. For example, for the cDNA, which we tested priming, producing the cDNA with random hexamers or with oligo-DT. We also tested different amounts of material. We tested using a single amplicon for the TELL-Seq processing. We tested a panel of amplicons. Overall, it can be summarized in these 58 tests. So I want to highlight the last two rows, those ones in red. So one of them refers to how many times actually we were able to recall the presence of the mutation or the single nucleotide variant. As it can be seen in the table, it's 100%, so it was 100% success in the 58 tests. The last row indicates how many times we faced those single nucleotide variants or those mutations as expected, as previously reported. And of those 58 tests, it was correct in 57% of the cases. So we almost got a 100% efficiency in terms of phasing. If we move to the next slide, which will be the last one. So the take-home messages here, I think, are very clear after what I just showed. But basically, we can confidently say that we can phase PCR products of different sizes. The smaller sizes that we faced in these studies was from 2.7 kilobases and the largest was 13 kilobases. And as I showed, it was successful in almost all the cases but one. Okay? So I have shown how we can actually phase a single product, and we can also phase a panel of products mixed together. It's important to annotate here that those PCR products, they cannot overlap. At this moment, it should be like two different regions in the genome, okay? Not different samples, but different regions in the genome in a pool. And then, as a reminder, we always say this is the TELL-Seq library prep kit is for research only, research use only. And then, as I mentioned before, what is the final user guide for PCR products will be officially released soon. It is not yet available because we are trying to finalize a few recommendations and a few technical tips. But actually, if a particular customer wants to have access to what we have right now, we can definitely do it. And then all the technical questions obviously can be addressed here, but in any case, it can be emailed to Andrew Amphora if it's after this talk. So that will be it for me, my presentation. So Andrew, if you want to take it back.
All right, great. Seeing some questions in the chat already. So please keep the questions coming. First question, can application number 2 be used with genomic DNA? So yes, we can. Okay, so a whole bunch of questions. So we presented phasing of targeted amplicons. We actually first rolled out phasing at the genome scale. So the amount of DNA needed, so the second question, how much DNA is needed to prepare the library? So we went over how much is needed for the amplicons. For the genomic DNA, it was also quickly on that slide. If you're looking at a larger genome, such as a human genome, we're talking 5 nanograms of DNA. If we're looking at a microbial whole genome, you'd be looking at about half a nanogram, sorry, 5 nanograms or half a nanogram of DNA for a microbial genome. Another question was, what's the max size of resolved haplotype? Tom, could you maybe take that one? What's the largest haplotype we've been able to phase so far?
It really depends on what that means. It means the individual targeted. Have we tried? Because at the whole genome sequencing level, for example, is we look at the MHC region. That's almost 4 megabases. We can phase it completely, and also is at whole genome sequencing level is we know our N50 phase block can be around a good time can be around or even about 40 megabases. So by average, it's probably around 10 megabases. So that's what we know. If the question really referred to for targeted region, so we already demonstrated with HLS catch method, we target extract 200 kb BRCA genes. Yeah, we can phase it completely. Yeah, so it depends on what's really the question means.
And yeah, it looks like there are
yeah, well, one more thing for those kinds of targeted application, so we usually just use a 0.1 nanogram DNA as the input. Yeah.
And just one more follow up on the targeted question, because I don't know the number since it's a new application. How many samples are we expecting to get for out of a standard kit for targeted? Is it 12 or is it going to be more than 12?
Yeah, so for current standard kit, we basically treat a targeted region as a small genome reaction. Okay. So by design, that's 12 reactions. However, for Amplicons specific based method, we do believe there's room. We actually can do more reaction with current kit. So it's not ready to roll out yet. So it's a minimum is 12 reactions. I think down the road, we probably can use SIMKit to do more reaction for the Amplicon application. Okay. And yeah. I saw that the previous question that they asked if they're able to capture region between 50 to 100 kb. Yeah, it will be perfect going into our reaction. Again, we need no more than 100 picogram, basically 0.1 nanogram, to do that kind of case.
And it looks like we have another one. So moving on to genome scale questions. Are we able to phase smaller genomes, such as a 9 megabase genome, assuming it's diploid?
Yeah, that'd be good to, in principle, it should. The things usually for us, the small genome is just this is a single chromosome, and yeah, we haven't had chance to do the deploy the small genome. But based on what we do on the human, yeah, we believe we can do it. Yeah.
Okay, so if it's small genomes and you're looking population scale, depending on how rare is, the dynamic range might affect the feasibility of that, just based on what we've done in the metagenomics space. So the question of the small parasite genome, looking for rare variants, that might be a question we could better follow up on via email. I think there'd be a lot of situational variables there in that I'm just thinking of off the top of my head. But the answer would be probably with some caveats. And just a reminder for those in the audience. Oh, let's see. I'll just mention one thing, then we can address the question that just came up. Feel free to ask us about anything from TELL-Seq if you had other questions outside of targeted. We do want this to be open questions, not just what was presented today. So if you're an existing user or we're thinking of a different application, we're happy to chat on that. So we have a question: what is appropriate software for phasing? Actually, I'm going to share a slide just because I have a diagram of that. That's a great question. For those of you not familiar with our workflow, I have a slide for that that will, let me see, can I, give me one moment. Okay. So there are, so the question was, what is appropriate software for phasing? What I am showing up here is our software workflow. So everything that's depicted in green are software tools produced by Universal Sequencing. In blue are legacy software items from 10x Genomics. Our data type are similar. If you've used linked reads with 10x Genomics and like that software, you can do that. But I'm going to talk about our workflow. So going from top to bottom on the green portion of this diagram, you will sequence your libraries, and coming off of the sequencer, regardless of what the downstream application is, you'll run our QC pipeline tool called TELL-Read. That will provide some QC metrics and do some cleanup of the sample, the molecule 18 base pair indexes. Once you have your cleaned up FASTQ files, then you can take it into several different pipelines. Our phasing tool is called TELL-Sort. So that would be what we would recommend for phasing your genome or targeted sequence. Our tools are, so all of our software are available as Docker or Singularity images, freely downloadable. We do also currently have TELL-Read and our de novo assembly pipeline TELL-Link available on BaseSpace. TELL-Sort will be available on BaseSpace in the near future. So we do provide free access to the software you would need for phasing your TELL-Seq samples. Going to stop sharing that, but I have other slides if there are other questions. And just what was I going to say? Oh, some more questions coming in. All right. So is high molecular DNA needed for the library prep? If you are doing a whole genome scale application, we recommend DNA greater than 40 kb in length. And actually, I have a slide that I'll share that kind of shows the relationship we've observed between DNA fragment size and observed phase block. So the recommendation for whole genome scale of 40 kb comes from observations like you are seeing here on this slide. If you're doing a whole genome application, we really recommend DNA fragments greater than 10 kb in length to see the most impact from this assay. Of course, if you're doing targeted sequencing, we can modify our protocol for targeted applications, as you've seen this morning. So really, we want ideally fragments greater than 40 kb. If you have a slightly shorter DNA sample, we provide suggestions for modifying the protocol. But really the best case scenario is going to be high molecular weight DNA for a whole genome application. And the only other thing for the high molecular weight DNA question also, obviously project to project, how contiguous is contiguous enough for your assembly or phase block will be variable. The 40 kb size is what we've observed to be achievable for most labs and gives great quality for most projects. Some projects are fine and get sufficient quality assemblies with DNA closer to 20 kb in length. Again, it's a little bit project specific as to where that 40 kb recommendation comes from. All right, we'll leave stuff open for another minute or two. Looks like questions might be running out of steam. Oh, here comes some more. Yeah, so a question about installing our software on a core computer. So if you have access to high performance compute environment at your institution, it should be installable. If there's a specific use case scenario, go ahead and email me or our tech support email address. I know we do see a lot of variability, but we've been able to get most compute systems up and running. There may be system specific concerns depending. Do you have any products that could pair with this kit to capture large regions? I also have a slide for that. Let me see, do I have it in this deck though? So I don't have it in the deck that I have handy. So the question is, do you have any products that could pair with this kit to capture a large DNA region? So we do have an application note up on our website where we were using a product called HLS Catch from Sage Bioscience, where and that's what we use to enrich the BRCA genes that are of that 200 kb-ish size that you mentioned. So you can either email me and I can share more details there, or you can find that application note on our website. That is something that's definitely feasible, that we have done some proof of principle studies and have an application note focusing on that specific use case of enriching for 100 to 200 kb regions of the genome. Let's see, and I saw there was a question just of somebody who joined late for an overview. We will be, as David mentioned in the chat, we will be sending out a link to this recording. Also, what we are trying to do with these Q&A sessions is every other week will be a little bit more of a general Q&A that will have a general overview of our core technology, and then we're trying to add in more applied examples like we did this week. We've seen a fair number of people joining every week, so we want to put in some variety there to make it worth your while to attend often, so you can learn in digestible chunks and we don't spend 2 hours bombarding you with too much data. Do we have any suggestions for a SV tool? So we do have an application note where we talk about SV analysis and present several analysis options. If that's not, I think that's been posted on our website. If not, it will be posted very soon. So take a look at our website. If you can't find it, go ahead and email me or email our tech support, and we can share that with you. I don't have data from that app note handy in my deck.
Yes, just a quick summary is for SV detection, there's currently indeed still challenge for the link rate. There's no one perfect tool for everything. So current approach what we've been doing is we use combination method. We actually combine both a short read SV tool and link read SV tools. So again, Andrew mentioned that app nodes. In that app nodes, we look into is some link read specific like neighbor linked SV, and then combine with short read specific SV like Manta and another tool together. So by doing all this combination, we can truly identify majority useful information from the link read. And also is there's traditionally attacks their own tools. It's called a long ranger. If you can also use convert our data to the tag comparable format, run a long ranger. So there's a variety of tools available. At this moment, we do realize, to get the most information out from the data, it's probably worthwhile to run multiple tools together rather than only rely on one tool.
All right, great. I think looks like questions have slowed down. So again, go ahead and email us if you think of anything. So if you have any questions, I would probably be one of the best to coordinate technical questions with. Looks like we have another question or kind of around wanting a general informatics talk. So David has shared my email in the chat. And I guess we will wrap up and leave it there. You guys have my email for technical questions if there's something we haven't gotten to. And thank you everyone for attending and your interest in universal sequencing and TELL-Seq.
Thank you.